Welcome, Tyler, to the Collaborative Research Center Origin and Function of Meta-Organisms. And it's very nice that you find a little bit time to talk behind the scenes, behind the daily work in the laboratory. You are here at Kiel University as a postdoctoral fellow, supported by a fellowship from the Alexander von Humboldt-Foundation. Just recently got your PhD and immediately then got this prestigious fellowship, and you came to Kiel at a very unfortunate time, of course. I mean, moving during pandemic times is challenging. But all the more, we are very happy to have you here, and you are making certainly an impact, and I'm very interested, and I guess numbers of our members and others will be interested to learn a little bit more how you experience these times and what drives you and how did you get to where you are right now. As I understand, you always were kind of, I see you as a kind of an ecologist, a marine ecologist who are interested in the deep biology, the networks of marine organisms, and we will talk about what you do recently. You just had a beautiful PNAS paper. Can you tell us a little bit about how you came to the field where you are right now?
So I had always had some interest in the ocean. I knew that I wanted to study marine animals. I took a broad undergrad degree in marine science, and the class I liked the most was invertebrate zoology. So I started studying and looking at touch tanks, and really the animals that stuck out for me were sea urchins. And I came to then find that they have these beautiful larvae. They have this unbelievable developmental program. And I just began to be really fascinated by how these larvae navigate their time in the ocean and find their way back to the seafloor. So I started studying how a bunch of different ocean processes influence larval development and larval survival. And one of the ones that I came across that I thought was a bit unexpected was that there were microbes inside their tissues. So I became quite curious if those microbes help the larvae while they're in the water column. There was only about 2 studies at the time, and there were both two different groups of researchers that were like one-off observations while they were looking under the microscope. So while I was studying ecology, I then saw these microbes and was curious how they interact. And then before we
continue and to this wonderful study, may I ask, was there something in school or family or something? Who drove you into that? I mean, who raised your interest in marine organisms?
So I grew up I was born in an ocean state in the US. And I moved to a land state that was big on skiing and nothing to do with the water. And I guess when I was young, I would always ask my dad to go down to the ocean. And then when we couldn't, he decided to put a fish tank in our house. And I always wanted to take care of it. So I just had this interest in it, and he fostered that.
Great. Let's jump and come to your latest success, and that is, I think, a wonderful PNAS paper, which still stays with the sea urchins, but you alluded already a little bit to it, and you were getting deeper and deeper in this larvae life and the biology of the larvae. Can you summarize in a few sentences, in kind of more general words, what's the major finding of that new paper?
Yeah, so one of the big evolutionary successes for Symbiosis has been animals having a gut. It's this really, I think using the word nice is probably a little too friendly, but it's a good environment to house microbes. So the main question of the paper was, what happens to this microbial community when the gut is lost? And what we found was that there is a reduction in the different types of microbes and how abundant they are when a host loses a gut. So it really fits into the idea that when animals evolved a gut, that these communities probably evolved with them.
Would you summarize, that's the message for the broader symbiosis community, the important going away from the larvae, the sea urchin larvae. So that's the importance of the paper, that it highlights that there is this distinct and specific correlation between a gut microbiome and the presence of a gut, of course.
Yeah, I would definitely think so.
This field we are working on, and I'm very lucky and happy, and so that the Humboldt-Foundation supported your coming to Kiel. This symbiosis research, it started with a very few people on very exotic animals, and it didn't really catch too much visibility in the broader biological field, and all was about genetics and development, etc. But now it became one of the major areas of activity, people trying to understand how, the fitness of an organism certainly depends to some degree on the symbiont community the organism is colonized by. Why is that? And this attracts more and more young students. And if we do, as we did, Gordon conferences, you were part of that, this is full of young people. What is so attractive for young people to enter that field?
I think symbiosis really and of course, by its definition, symbiosis really ties together a lot of concepts that maybe once didn't seem as tightly knitted as they're now appearing to be. Whether that is just a simple relationship between an animal and relationship between an animal and a microbe, or between different disciplines, both ecology and evolution and development. Viewing it in a microbial light seems to allow a young researcher to look at a question from all these different angles, and in the view of symbiosis, they appear to be a lot more connected.
Would you agree with the thinking that biology in the last 50 years got very reductionistic and type of silo thinking, with the big advantage that we understand a lot in depth? But the real biologist really has the wants to have a kind of what I call a holistic understanding of the whole organism in its environment, and when now this is possible again, that is of course then an attractive field to enter.
I agree completely. Naturalists used to be really common in departments all over the world, and departments and researchers rarely are just naturalists. And they've really been replaced by model systems. So definitely with the exploration for new symbiotic relationships comes with people stepping back out of the lab and going into the field. For the sea urchins that I've been working with, people have used them before, but just in a completely different context. They haven't been trying to culture them as close as they can to in the ocean as possible. So we definitely are seeing that a bit.
Well, you mentioned the word model organism. I sometimes think by myself, what is a model organism? You remember a funny discussion at the Gordon conference. A model is a non-model, a non-model, non-model. What is a model? What is a model organism? You are studying sea urchins. Is that a model organism? And then, if so, a model for what? And what does it need to be a model organism?
Yeah, I think it's a relative comparison. First, what is the definition of a model organism? Something where you can use just about any possible technique available to study its biology, I think is probably a very broad definition. Sea urchins have definitely been viewed as a model system in a developmental context. They have for a while. They're easy to use in the lab and to manipulate. They're definitely not from a symbiosis perspective, but it probably won't take that much to get it to, that status. And that's simply because people have been working on them for 150 years. They were very easy to go down to the seashore, pick them out and spawn them to look at their gametes.
Yeah, I think I would even go a step further. And so sea urchins are certainly a well-established model, and, the regulatory network, the developmental biology, Eric Davidson, that's sea urchin. But go back and go to the sea and do a scuba diving or something, and you find something interesting. Couldn't modern technology allow you now, when you have a real good question and as a naturalist, this organism may inform me about that, wouldn't that available technology allow you that you really study that strange organism in depth, and by that then you make that organism to a model organism?
I think that's, yes, certainly. It seems like some of the techniques that are at least being used right now in symbiosis research, the one that of course comes to mind is any type of sequencing should allow you to really advance what is known about that system, to then do strict empirical work that is often done on model systems. So definitely having these broad, very easily adapted techniques should allow non-model systems to become model systems, or be very experimentally tractable.
Yeah, I think the fascination of symbiosis research partly is also due to the fact that really, because of the modern technology, we can, maybe not all, but many different organisms can now be used really in depth at a mechanistic level. I think that's what we all agree we need. We need move away from correlation and associations to get an understanding of a causality. But this does not mean that you all have to be as good as Drosophila or C. elegans in terms of technical aspects and accessibility. But these many different organisms may tell you so much about how nature managed to make so many fit organisms. And I think this is really fascinating, which was not possible 30 years ago, as simple as that, because there was not the means to that. The second aspect which contributes to that, and you said it right at the beginning, is that you confronted or you compared the naturalist and with the, whatever you call it, experimental biologist, molecular biologist, reductionist, whatever. But this also seems to me to reflect an old and overcome culture. And your work certainly crosses many different boundaries and crosses departments. And are you an ecologist? Are you a microbiologist? Are you a biochemist? Are you?
I have no idea.
Yeah, but the question would be, as a junior investigator in the field, how important and how challenging is interdisciplinary working? And is that something which you really think is bringing the field forward?
My undergrad degree was in marine science, and it was this group of aquaculture folks and marine molecular ecologists and oceanographers, and you're always going back and forth. And that was what really spurred a lot of ideas. And I was in a broad biology department for my PhD as well. And other researchers have a completely different perspective of something that you may not realize you needed to know, or that may provide some hint at the question that you're just sitting on. And that definitely came to fruition in this recent paper. And once it does, I think that's when really cool findings can come up.
Yeah. Related to that, I mean, you are now made a very tough decision, and during pandemic times, you left the US and came to Europe, came to Germany, came to Kiel, and joined a new institution, the Geomar Helmholtz Center. In your experience now, what does it need that an institute has to offer in terms of structures, platforms, communications, etc.? What do you expect from an institution before you choose to go there?
So I will precede my answer with saying that I got a lot of help, and that there are a lot of people who made it possible. You and Ute being probably the two most important ones. But the thing that I really struggled with when I got here was knowing what is needed to become a resident. It's on top of figuring out where the pipettes are in the lab and where I can find my animal in the ocean, but knowing what legally makes you allowed to stay where you are and work and find just the normal resources that were easy to find before.
You talk about bureaucracy and so immigration offices and stuff like that?
Yeah, and how to pay your electric bill. Usually it's not an issue of money or timing, it's who do I send this money to? How do I send it? There was a lot of things that I learned very quickly of what not to do that was fine in the US. And just knowing the in and outs would probably be something that would be really helpful, because doing that on top of the day-to-day of trying to maintain some research program is quite challenging.
Yeah. German universities, and in particular also Kiel University, we had a talk or a meeting yesterday about that, has a deficit in terms of internationalization. And I think this is exactly one of the problems where we have to make it more easier for our international guests, which are all invited and most welcome and highly recognized. But if they fail in filling out some very trivial daily forms or something, because there's something wrong, and we have to change that, and we have to improve that. I mean, it's clear you go in a country where English is not the native language, but you also go to an academic institution where English is the lingua franca. And so I think that's why I ask this type of questions. And this is an interesting point what you raised, which goes away. I thought about scientific structures and platforms. And I think many institutions like the Geomar are very well structured in that respect. But if you have a very hard daily life in terms of surviving, as you say, to pay your electricity bill, that is something, but there is room for improvement. I fully agree. Yeah.
Just one note on that. I was very close to a multi-thousand euro issue because I, without having any idea, signed up for the wrong healthcare. It is a common one in Europe. But it was not the correct one based on my contract and my financial position for being here. And then it was illegal to be out of it without signing up for something new. So avoiding those situations, I think, would be pretty good for future people. Yeah.
Well, you made it. You're pretty successful, and you'll continue to be successful. And there's no doubt that you are on a uprising path towards a very international visible career. If you look back, and if you now maybe talk to the very junior, the undergraduate members of which are somehow associated with our collaborative research center, is there anything would you like to tell them? What is necessary and what they have to stick to, and what they have maybe to abandon and to forget to make it to that nice and high level where you are right now?
So there's two things that come to mind. The first is just try a lot of stuff. I started off actually in aquaculture. I was working with the sea urchins there because it was the only thing that they had, but I cultured clownfish actually for a summer because I was interested in the ocean, and worked on diseases and biomechanics for a little bit of just trying to see what I actually liked. So my first piece of advice would be just to try a lot. And the second is that failure is totally okay. And I think anyone at any degree of seniority would say that failure is the expectation, not success. And it shouldn't say anything about your quality to be a researcher or a scientist or naturalist or whatever you want to be. So those are the two that really come to mind.
Thank you very much. Yeah, I mean, continuing on that, science is a communication business. Talking to each other, meeting each other, getting ideas, spontaneous ideas, not in a pre-planned meeting which has topics which then be discussed, and then the discussion leader says it's over now, so see you next Monday. And we are living now in times where science is isolated. Scientists are isolated. Physically, we talk online under, very controlled conditions. Spontaneity is rather rare. I mean, it can happen, but how do you cope with that physical social distance time now? You do quite well. I see you still smiling and shining eyes and burning of getting to the next step of research and career. And so how to cope with that certainly very challenging time?
Yeah, probably the best way that I cope is actually go running. It's to actually not do science. But I think what may be more beneficial for people broadly is that I try not to judge myself for not being as productive. I know there's a degree of productivity that I could be achieving and I want to achieve, but just realize that you're just in a circumstance where it's probably not possible, and it's totally okay. And I still try to be creative. It just takes a lot more effort and a lot more time than normally.
I fully agree. I think we should really stress that this time is not easy for being creative. I mean, it needs a lot of self-discipline. This has many benefits, of course, also, having more time, being less disturbed. But when we talk about creative scientific thinking, we realize that our sources for that creativity come from many different directions, and which are usually very hard to define. But when they are cut off, right now, then we are missing them. So yeah, it's true. You're quite established already in the field, and you write perspectives and about going beyond your own observations. So coming to an end of this little chat, but I want to ask you, where do you see the field of symbiosis research, or maybe marine symbiosis research, in the next 5 or 10 years? What are major open questions, and which questions you think will be answered in 5 or 10 years?
Oh boy. I think the major questions are probably stuff that we didn't realize was influenced by microbes that actually are. The thing that comes to mind for me are major life history transitions, where we thought it was this host-directed process that may not be. One of the big takeaways from this recent paper was that a big change in development could have been by a reproductive manipulating bacterium, and had maybe much less to do with the host. And those types of changes in animal lifestyle are found all over the place.
Mm-hmm.
So I would not be surprised if looking at major transitions in an animal's life outside of the life cycle would be one area where these types of studies really blossom. Another one that I think about are all these lifestyles that people thought were quirky. Like, how does an animal live like that? There are larvae that people have observed that can be in the larval phase for years, when usually the normal time is a few days to a couple weeks. Yet they just hang out and have no worry about being in that state. So I just wonder if there is this microbial influence that allows them to sustain these really intriguing lifestyles.
Yeah, thank you very much. I think this is two actually pretty novel and original ideas or visions in the future. This is I fully agree. What I want to add is, and you are good in that, and not everybody is good in that, is it needs also a lot of effort to communicate that findings in our classical communities, the developmental biologists, the microbiologists quickly learned that microbes are not exclusively pathogens. And now, so these are now great members and friends and colleagues and experts which we need because they know so much about microbes. But having a good deal of communication skills, I think, is necessary, because still what you find, and just the last few conclusions, the sentences which you said, that you expect that we discover more and more key developmental processes and transition steps, which may be influenced much more by the microbial environment than by the host itself. That is something which a classical developmental biologist still has hard time to accept, in my experience, and it needs careful, persistent, and trained communication to convince the community. Would you agree to that?
100%, and I think that will dissolve with time, just as more data that they can look at with their eyes comes out, instead of a researcher like you and I trying to hit them over the head with it. I think it'll be inevitable that they'll have to accept that complementary perspective on that aspect of animal biology or plant biology.
Yeah. Where do you want to be in 5 years?
I think by then I would like to be, or have, a lab that is beginning to be pretty independent and, at least to some degree, self-sustainable. I would love to have the transition where I can be in a position to seek funding for projects that I'm curious about, and truly curious about, and have be in a network of people in students to work on these questions, and hopefully more that aren't even in my head, come up from all the people that I'm working around.
I'm very confident, Tyler, that you are there where you want to be. In 5 years, you are already very independent. I remember the evaluation of your thesis by your thesis advisor, and your thesis, you were already pretty independent and your own ideas, and it's great. So you can count on us, and I think we'll support you very much on your future steps. I wish you all the very best. We come to an end of this short chat, but it was a great pleasure, and thank you so much for sharing personal views, which other members of the center, junior members, but also senior members may be interested in learning, and it may also help some of them to go through challenging times which you have managed somehow to go through. And so thank you so much, and very best wishes for the future, and I'm very happy to interact with you as much as personal as possible, and beyond the online format. Thank you so much.
Thank you for having me. It's been a pleasure to sit down and talk.